Hi, hello. Welcome to Tea Time with Tocklai. Today we will talk about RNAi, which Tocklai has been working very actively on. To talk about this, I have invited Dr. Rakesh Vijay Kumar, who has recently joined in entomology team under Dr. Somnath Roy. To tell more about him, he also holds a gold medal from IARI for his PhD. Welcome, Dr. Rakesh.
Thank you, sir. Thank you very much for this opportunity.
Rakesh, let's talk about RNAi. What is it basically? Can you explain in simple terms that every planter and small growers can understand?
Sure, sir. RNA interference, which is also called as RNAi or gene silencing. This is a technology where we will silence or turn off the gene from making it to perform its essential function. Like that, we can prevent its normal functioning.
Okay. When you do this, it will not harm the tea plant and the tea genomics and tea genetics?
Sure, sir. This is like a much needed question.
Okay.
So the technology is like that. It doesn't have any role to modify the DNA of particular plant or even the particular insect.
Okay.
So we are not going for any modification of the particular gene. Instead, we are just normally using that as a spray-based application, like normal insecticides that will attack the particular insect without harming its normal genome organization, including tea plant.
Okay. How does it really work?
Sure, sir. Actually, like all the living organism, all in the sense animals, human beings, plants, in short, we can keep it as eukaryotes. All eukaryotes, this RNAi mechanism is conserved within their body.
Okay.
So like this is generally they have discovered in 1998 as a viral defense strategy. So when a dsRNA virus is encountered, then our body mechanism will activate and silence that. So this is the mechanism behind it. So to activate that particular pathway, we need a triggerer molecule that is called double-stranded RNA.
Okay.
Once this double-stranded RNA is encountered with the particular cell, then the normal machinery starts, where our cell, it will identify the dsRNA molecule, then that will be chopped out by certain enzymatic mechanisms within the cell, and this will go and bind against target mRNA of a specific gene, and it will degrade it from producing the protein. This is the actual mechanism.
Okay. When you talk about double-stranded RNA, is it readily available or you have to make it for a particular pest and insect?
Sir, it is like we should synthesize it for particular pest and the particular gene we are targeting for that pest.
Okay. So in actual sense, you need to take the pest and then from that you extract the gene and then start doing this work?
Sure, sir. First, we want to identify the target pest, and from that pest, we will identify the target gene.
Okay.
Which gene we want to target. For example, we want to target a gene responsible for its reproductive potential. Then we should take that gene and we should collect the sequence and we should start synthesizing the double-stranded RNA.
Fantastic. Sure. Will there be any genetic difference within a particular organism that will impact your RNAi development?
There are chances for like this modification. So what we used to take is that we used to collect the genome from the data bank, and then we used to design that particular stretch which is conserved, so that it can act against that particular species.
Okay. Okay, now when it comes to real field applications,
sure,
how this technology can work in vast area like tea plantations?
Sure, sir. This is much needed question. Like actually this particular technology, when it was first identified, this was used to study the gene function. Then later it got updated in the pest management point of view. So in that case, there are two strategies. One is host-induced gene silencing and spray-induced gene silencing. Host-induced in the sense, like we all might be aware of the genetic like modified plants,
Yes.
BT cotton, like these kind of plants. So we will engineer that particular gene, dsRNA production gene of the target in the plants to synthesize the dsRNA. But these have limitations. In our Indian condition, adoption of this GM crops have certain limitations. So the another viable strategy will be the spray-induced gene silencing.
Okay.
So we will make this dsRNA as like sprayable insecticides and will be spraying on the plants. So this, in case of tea plantations, when we are coming, this is again a question mark, because the plantation is on large areas. So that much capability to upscale the dsRNA production is still under process. So we need to work on the production of such a large quantity of dsRNA. Actually, what we have earlier worked when in my institute was on small scale. That itself, like, we couldn't scale up. So we are in the process of scaling up of that production.
Can you also tell us about the status of this technology in the real field application, worldwide probably?
Yes, sir. So worldwide, like recently means in two years back, 2023, it was the first product which came into existence. It was in USA, targeting a beetle, Colorado
So
potato.
it's a very new technology.
Yes, sir. It is very new. It is in nascent stage. We can say it is almost in its nascent stage, and it is emerging.
Okay.
So this has been recently, the first dsRNA based pesticide was introduced in the field during the year 2023 against a beetle in potato plant, and that product is called Leadprona by Greenlight Bioscience as normal dsRNA spray based insecticide.
And is it working on the field?
Yes, sir. It is for field level application. Yes, it was also registered. Their regulations is monitored by EPA, Environmental Protection Agency, and it has been registered by them for a period of 3 years, and it is being used.
Yeah, then it throws another question here. Like EPA is there for years.
Yes.
What is there for us? Like whether the same CIBRC will take it up, or we need to go for different kind of regulation?
Yes, sir. That is still a debatable question. Like actually, if we see about what is this dsRNA actually is, compared to other international country regulation, they are saying it as biological pesticide or biopesticide, because we are using the molecule of biological origin, the double-stranded RNA. So in that condition, in our Indian perspective, say we don't have any product till date. So like if we are coming up with a product, and if we are claiming it under as a biological pesticide, then ultimately it should go under CIBRC guidelines. And for CIBRC guidelines, to get it approved, we need 2 to 3 years data under multi-location trial. This is the current scenario.
Coming back to our own research, planters have been hearing this for the past couple of years.
Sure.
During the presentations, that's what I heard from the planters, that they have been hearing this.
Sure.
What is the status of our current research and what organism we are trying?
Sure, sir. Actually, like I have joined here a couple of months back, and it is interesting to know that already one project is there with TRA under collaboration with University of Calcutta under the sponsorship of NTRF.
Correct.
So they are working on this Helopeltis, where they have identified the resistant and susceptible population, and they have identified the genes responsible for resistant in that particular pest. And now we are in the stage like we have identified the resistant genes, and now we will develop the dsRNA for those genes, and we will be taking it further for laboratory level validation and field level application.
Yeah. Thanks to NTRF for sponsoring such a innovative work, right? As you said, like it is only 2 years old in US.
Sure.
Means we are going very, very ahead in time.
Yes, sure.
So NTRF has come handy on time.
Definitely.
To sponsor this project. Now the technology has been used in US with the registration in EPA. There should be some kind of feedback coming from them as what are the difficulties in really making this product viable on the field.
Sure.
Can you postulate some difficulties that we will be facing in the future in tea field?
Sure, sir. Actually, these double-stranded RNA molecules itself, like they are not that much stable.
Okay.
As these are biological molecule, it will be having a short shelf life in case of naked double-stranded RNA molecule.
Okay.
So to make it available throughout the system and to make it stable and protect it from external environment, there are certain researches being taken up by conjugating it with nanomaterial or some biopolymers, so that will make it available throughout the plant system and reduce its instability.
Yeah.
So this is one of the technology. Now currently it is being worked on. Like in the case like as in a case we take a normal dsRNA and we are spraying it, its stability will be there for short period. And by conjugating it with a biomaterial or any other cargo molecule, so it will be having an extended bioavailability.
Yeah.
So this could be one of the things which should be taken to extend its bioavailability and its persistency in case of real field application.
In these 2 months, have you started working with any other organism using RNAi technology?
Sure, sir. Like actually, like this is a very customizable platform. At the same time, we can take multiple pests.
Okay.
So like now currently like we are in identifying the target gene for Helopeltis, and again like there is the recent invasion by a recent attack by this thrips and green fly. So we are targeting those two pests also in this, bringing under the same umbrella for a combined protection or integrated protection.
Yeah. I think this kind of studies are very important because it can bring down the MRL to zero. There is no MRL left on this because there's no chemical and there's no pesticide involved in it.
Sure.
But at the same time, because being a new technology, how do you postulate that how many years it will take for us to see some real molecule application on the field? Double-stranded molecule application I'm talking about.
Sure. Definitely, sir. Actually, like if we see, like the product can be made because as we have already known the technology, it can be made. But the question here is how it will be performing in tea plant system.
Correct.
Because like tea is a perennial bush and we are maintaining it, and how this actually performs in the tea plant system and what happens to the essential biochemical properties of the tea once after spraying. So these needs to be validated. So it takes minimum of 2 to 3 years to study all these properties and to come up with a product.
Okay. When you want to come up with a product, do you need the help of some manufacturing agencies, or it is as simple as you can make it in your laboratory and then sell it?
This is most needed question.
Yeah.
Because in laboratory scale though, definitely we can't produce that much quantity.
Yes.
So definitely we need an industrial collaboration to upscale this particular dsRNA production, which is needed for its large scale application.
Okay. So we need to find an industrial partner once the technology is ready with us.
Definitely, sir.
Okay. Maybe a greedy question from a planter, like, can this technology be used for multitasking? Here I mean that in one application, you can go with pest as well as disease. Is that possible?
Definitely, sir. Using this technology, it is possible because we are targeting a particular specific gene of particular pest.
Okay.
Even we can target more number of pests by taking the specific genes of the particular pest, including the diseases, what we want to control.
Yeah.
So it can be a cocktail formulation where one application targeting multiple pests and diseases.
That's really quite interesting, actually. With that, let us conclude this podcast. Thank you so much for coming to this show and then explaining what is RNAi. Thank you.
Thank you very much, sir.